proteomes, and moving forward.
Okay. Am I on? Sorry.
I can't neither see you nor your presentation as yet.
Would you like to share your presentation, or do you want me to share it from this end?
I'm sharing it. I have now.
Oh, yeah,
So
got you.
you
There
see
you
that? Is that okay?
Yep.
Whoops, on the conclusion slide. Okay, so I'd like very much to thank God for his Herculean efforts in pulling all of this together, and it's a wonderful thing that I think he's managed to put together. So I'll try not to let the slide down too much. So what I really want to talk about, really very, very briefly, is mostly published work, but is a little bit of an overview of what we've perhaps done in collaboration, mainly with folks at Julius Lukash's institute in Český Budějovice. I'm interested in very much, and I've always been very much interested in the molecular cell biology of organisms, and I think we had a comment coming up, or a question, as to how closely euglenids are related to trypanosomes. And I've ripped this off from one of Julius's recent articles, and here's my sort of favourite usual workhorse, the African trypanosome. It's favourite workhorse because it's easy to work with. We can do all sorts of things with it, and so we can learn quite a lot about its biology. And Euglena is over here, but this is this particular clade is a sort of tripod. There's three separate clades, which also includes the diplonemeids, for which we now have 2 or 3 genomes sequenced to a reasonable level of quality. Now, one of the problems that, when thank God was doing his PhD with me in the lab, that we kind of came across was that the Euglena genome is enormous. Unfortunately, as we sort of started this a long time ago, every time we did an assembly and added more data, essentially the goalpost kept moving, and we basically got a larger and larger estimate for the genome size. So we think it's probably around 2 gigabases. I think that's probably a reasonable estimate. It's consistent, certainly, with many earlier estimates that placed this at around about a gigabase or so. Which means that we are dealing here with a genome, at least in terms of its just raw size, of similar complexity to the human genome. And one of the things that really came out from, thank God's, work in this area was that an enormous amount of the sequence was lost essentially into the bin due to high repetitive nature of that sequence. It was very, very hard for us to put together large contigs. And I think, as a previous speaker pointed out, I think Harris, indeed, the N50 number is not particularly appealing. So certainly the way we were going about this and the technology using Illumina, and in fact originally 454, really didn't do it for us. It did allow us to put together what we believe is a reasonably accurate picture of the open reading frames and hence the proteins that are encoded. So I'm just going to highlight one experiment from thank God's work, which I think is quite important perhaps for people to appreciate. And the University of Dundee likes to pride itself on having access to high technology. So essentially what we wanted to look at was light and dark adapted Euglena. And so we used the high-tech solution of sticking cultures essentially into a cardboard box and having those sitting on the bench next to those under a light source, and essentially able to demonstrate that we had clearly lost the photosynthetic pigments. These were now sort of glass-like. Spectroscopy confirms that. They were growing reasonably well, but slower, etc. And then when we compared what we could see from a transcriptome with a proteome, we were really rather surprised that there is almost no correlation. The transcriptome really does not give us an accurate reflection of what we're seeing in the proteome. And so this is from a light-dark experiment. P-values are reasonable. There are a number of things, for example, that are essentially switched on only in the light or switched on only in the dark. but these are not reflected in what we're seeing in the transcriptome. So I think it's an important point to take on board. This is only one experiment. So it may be that under different conditions or under different or differential conditions that this won't hold true, but it is a cause for concern, I think, for transcriptomics. What it has allowed us to do, I say in collaboration with Julius Lukesh and Mike Hammond, who I think is talking later or tomorrow, and Vlada Hampel, it's allowed us to, for example, look at the relationship between the kinetoplastids, the euglenids, and the diplonema. So interestingly, one thing I think is really very interesting is an independent development of unique RNA editing pathways in the diplonemins and the kinetoplastids. We know that now because the euglenids do not have any evidence of the RNA editing machinery that has been well characterized in trypanosomes, for example. It's also allowed us to predict organella proteomes, and it's also allowed us to reconstruct some really rather frighteningly large protein families. So, for example, this particular protein family, it's clear that there are huge numbers of expansions that are specific to Euglena that are not being represented in other taxa. So, you know, this is really a very complex organism, and I think this also raises some issues with understanding function. It is very easy to take apart, and so we can isolate peroxisomes, chloroplasts, and mitochondria, for example, and we can show very nicely the top right that the mitochondrial genome, mitochondrial proteome is very nicely segregated away from the rest of the cell in this volcano plot, for example. And in the lower, you can see that the chloroplast and the mitochondria are also nicely segregated. So we can do that. We are working on one final proteome, which is essentially the pellicle and the flagellum. We have the data. We're still working on this, but hopefully you can see that we're able to make nice flagellae and pellicle fragments using various centrifugation techniques. So I'm conscious that we're running short of time, so I just really wanted to highlight a number of things that certainly are on my mind in terms of going forward, that we have potentially a massive amount of junk DNA, whatever that means, and that will certainly be a challenge for both improving the gracilis genome and any other euglena that folks feel is of interest. But what we have got so far is that it has given us insights into the evolution of parasitism. I highlighted RNA editing, and a large number of other things have also been nice and easy to dissect. So in particular, organelle proteomes, having the full nuclear orthome as well as a proteome has allowed us to make, you know, high quality predictions of the compositions of those organelles. I think massive gene families is going to be a big challenge. This has been a challenge in trypanosomatic research as well. So I'm, you know, really very familiar with that and the problems that that can cause. Certainly from my perspective, I'm concerned at the utility of transcriptomics, but I'm aware that proteomics is much more expensive and is technically rather more demanding than transcriptomics, which is comparatively straightforward. And a lack of reliable, reproducible transfection system, although I'm hoping to, over the next 2 days, to hear some updates on that, because I appreciate there's been some advances in some CRISPR technology for it and transfection, for example. So that's pretty much all from me. I'd just like to really highlight the folks that were responsible from this. So thank God who really took the lead in putting the transcriptome genome together. Where are we? Julius Lukesh, important for very many of our proteome analyses and comparative analyses. Not on here is Vlada Hampel for the chloroplast analysis, which was certainly the informatics was mainly done in his lab. And also Martin Zoltner, who really did much of the proteomics analysis and data processing. Okay, I think that's it. Not sure how
Thank you, Mark.